Hi, I'm Gemma. I'm talking about PFAS movement through soil and saprolite. Saprolite is highly weathered rock that has not yet become actual soil. And so this is how we did the sampling. First of all, we did percussive drilling at a sort of background site. So it's a sandstone hill slope in Shropshire in the UK. We got 24 core samples, but the experiments I'm talking about today are just based on samples from a soil pit. We took them in sort of 3 strips down this soil pit from 10 centimeters below the ground surface down to 1.2 meters below, as you can see on the right here. And to measure the background PFAS concentrations in those, we did solvent extraction and solid phase extraction, and then target analysis for PFOS and PFOA. And these are the results from that. So the sort of topsoil concentrations that you see there are sort of in line with similar background sites. And then there's a really sort of sharp decrease from 10 centimeters to 40 centimeters and deeper. And another interesting thing that we saw was that for both PFOS and PFOA, you have a shift from linear isomers to branched isomers as you go down the profile. And there are a few reasons that could explain that. So one is just maybe the branch ones transport more quickly through the profile. 2 is maybe you have this shift in the manufacturing method for PFAS in the early 2000s from electrochemical fluorination to fluorotelomerization, and the first produces a mixture of linear and branch, and the second produces linear. And so it might just be that the sort of older PFAS who have had more time to travel deeper into the profile were made by that earlier method. And then the third would be maybe there's some transformation from linear to branched as you go down the profile. And so the next thing we did was this kinetic adsorption batch experiment, and we used 10 centimeters, 70 centimeters, and 120 centimeters below the ground surface. We were just using a 0.01 molar electrolyte solution and spiking to do 100 microgram per litre initial concentration, shaking it and taking aliquots over a period of 73 hours since the spike. And because it's using the same samples as we had measured the sort of background levels, so we know what we're starting from in terms of the background level already. And this is how the results look from that. So you see more partitioning to the solid soil or saprolite in the shallower profile. We have, something going on with our no matrix spiked electrolyte controls here, because you can see that their recovery is actually worse basically than the rest. And so we don't correct the worst. We don't correct the main sample's recoveries based on those controls, because I think the recovery is actually different, and there's a few reasons that could be. Because when you have your sort of electrolyte only samples, you actually have lower pH and lower ionic strength, and so you're probably getting more loss of your PFAS because it's absorbed on the sort of internal wall of your plastic tube and sort of on the internal wall of your pipette tips as well. And also for PFOS as well, slightly strange thing from the time step 1 to 2, where you don't really expect it to be sort of increasing for some of them. But we think that's just, it's not initially well mixed enough in the first time step. But yeah, you see more absorption for PFOS than PFOA. And the next sort of things we've done as well is sort of measure, like you said, organic carbon is a really important parameter. And so we have measured the total organic carbon for the samples, things, as well as other parameters that we're looking at. But when you do the organic carbon and you work out the sort of organic carbon water coefficient, adsorption coefficient, it's actually lower for the shallower samples. So there is other adsorption that's more important deeper, and the sort of importance of the organic carbon is potentially less as you go deeper. But there are also different types of organic carbon and lots of other things I can talk about. And next, we're doing more like column experiments with the whole 1 meter cores that are like intact, which is the benefit of the percussive drilling. And then other things that we're analysing, like particle size distribution and doing ICP-MS for the actual composition as well. But I think I'm out of time, so thank you very much.